for help without draining you all. But anyways, so let me know it's 10, okay? Let me share the slide. It's not to scare you, but just don't get scared with the first slide. Okay. Looks a lot, but we'll deal with it. Okay, is it 10, everyone? Yes? Yes, it's 10. Yes, I will share these slides with you. Absolutely. Also, so everybody is good, right? So good morning, everyone, and hope you all are doing well. I know it's a lot of lectures, but believe me, I'll come outside class help. I'll do the Zoom and do as you were doing in the exam one. You all had a great average. All you need to keep an eye on is the number of lectures. I told students if you want to be successful, you have to assume that at least these 6 lectures are on the top up to carbohydrate digestion. Test is on Monday, right? Does that help you? I think you got to get over with these 6 lectures. You cannot carry them over in the next week. Unfortunately, next week's is 7 lectures. I will help you the same way I am helping. I'm sure you all will do well. Just keep an eye on your number of lectures, please. I will give you a focus. Yes? Somebody has a question already? I have posted the post discussion slides. If it helps you, my annotations, my diagrams, my overview, make use of it. So let's start the exam recap for the first 6 lectures. See that we talked till carbohydrate digestion and absorption. Please know one thing you do for me when you come next week for the session, please just draw glycolysis outline and TC outline for me. Please do these 2 outlines for me. Gluconeogenesis, HMP, other metabolism is only few steps which are clinically related, but I hope you can do TCA and glycolysis in and out. So can you please draw that outline for me so I can tell you which steps to focus on? Please, lectures are your heart of testing. Don't go to primers and CV before lectures. Primers are for you to refer. If you're doing lecture, you don't follow the basics, you lean back towards the primer. But you should start, your heart of testing will be your lectures. Now you have 44 questions. We are trying to give a little more because we have 14 lectures. We will see how it pans out and will we be able to do. Right now, we have 44 questions, maybe 1 or 2 more if we have room. Otherwise, there are 44 questions. What does that leave to? Maximum 3 questions or 4 questions per lecture. So that is why my focus is very important. Out of the 4 CV, at least 2 will be tested. We can't test all the 4 CV because there are only 44 questions, but at least 2 CV will be tested out of the 4. So it's best for you to read the CV and their questions. Please stick to the learning objectives. You can stop me and ask questions because that is my point for, especially for today. Next week, when I come to Zoom, students are more in panic. So I'll keep questions to the last. But today you can stop and ask me if you don't understand anything or you want me to repeat. Does that sound good? Are you following my pace? Everyone. Everyone is good?
Yes.
Okay, great. Let's go ahead. So what do you need to know about catabolism of and anabolism? Instead of focusing on the differences, I want you all to understand the concept when anabolism is turned on. And sorry, is it under foundation exam 2? What do you mean by Carlos? What's your question? Are you talking about the slides, the present slides? Carlos. What's your
Somebody was just asking where to find it, and just letting her know that it's under our modules. So it's found under the module named Foundation Exam number 2.
You mean my present file, the PowerPoint that I am showing now?
Yes, it's under
No,
Foundation Exam
no.
number 2 review.
No, it's not there. The exam files are across campus files. They are other campuses post it or not, but the team makes it. So the exam file, topic list, and disease list there is team approved. This I bring for from students' point of view where they can go wrong. So I will post this to your class rep as the outside class help. So this is not posted yet, Carlos. That's why I want to clarify. Okay.
Got it. Thank you.
Right. And also, just to let you know, you might have Rahul here and Teresa. They are doing a great job. And also, thank you for Shiva and Diego. They really did a great job too. And they did a voluntary work actually. And it was really great, worked out with Foundation 1. And I'm sure it will work out for Foundation 2. Please concentrate on the folder that they will make now for Foundation Exam 2, which will have all these files, right? This is for you to use as per your uh, convenience. If you benefit from my line diagrams, why not do it? If my overview gives you better grades, why not do them, right? Does that make sense? Otherwise, you have professor lecture slides. So instead of knowing catabolism and anabolism differences, I want you to understand that well-fed state is not just high insulin state. Well-fed state is high insulin and high ATP state. That is when body switches from, are you seeing my slides, by the way, everyone? Are you seeing my slides? Yes, that's the point. Yes, sorry, no
No,
question.
we did. Yes, we could see. I'm sorry.
Okay, no worries. No, sorry about it. I asked you, right? So, no, understand that well-fed state is not just high insulin state, it's high insulin as well as high ATP. When in a well-fed state, you're in a high insulin and high ATP state, body switches from catabolism to anabolism. So you must understand anabolism has some requirements. You will be tested on regulation of pathways because that's how you will understand normal in first year. So you can, sorry, relate to abnormal in the second year. So you must understand that there are some important regulators of the pathway coming up. What is coming up? Glycogen synthesis, for example. So synthesis is coming up. So you must understand anabolism has requirements. It requires what? Acetyl-CoA, ATP, NADPH, but most significantly, it requires insulin. So that is why who gives acetyl-CoA and ATP for anabolism? Burning carbs. That's why carbs may be fattening. Does that make sense? So you have to make the distinction of, am I in just a high insulin state? Or am I in high insulin and high ATP state? That's the distinction between catabolism and anabolism, and body wants to switch to anabolism. Understand that both of them are multi-step enzyme catalyzed pathway. You should know rate limiting enzyme or committed step of a pathway, every pathway that I'll bring in the next week. But don't forget that coenzymes help the enzyme. You must be familiar with, please, every vitamin and its coenzyme. Because if you are tested on vitamin deficiency, for example, if I tell you that there's a patient with niacin deficiency, which complex will be inhibited? If there's a patient with niacin deficiency, which complex will predominantly be inhibited? Complex 1, because complex 1 is linked to the substrate that are linked to NAD. Does that make sense? So you must recognize your vitamin and your coenzymes. Absolutely do that for me. Keep making the list. Now, instead of again focusing on what is the difference between rate limited and committed step, we have already done in the interactive session. Understand that every pathway I bring in will have one or two rate limiting or committed step. They are the bottleneck of the pathway. They are irreversible and they are subjected to regulation. There is long term versus short term. Understand that which are messengers. So now the key about getting good grades is, so how do you get good grades? First, you master each pathway, each lecture. So you bake the cake. Now, what is the icing, especially for foundation exam 2? The little that you integrate. If you keep the lectures isolated, please, then you will not be able to answer integration question. It's not mega integration, so don't stress out. Mega integration comes in your spring 2026. Right now, we are concentrating on little integration. So that little integration I will show you. For example, can you connect long-term regulation, which is at the level of gene, with signal transduction? Which messengers, which chemical messengers do you think would mainly or predominantly do or work at the level of gene, which is this regulation? Lipophilic or hydrophilic? Lipophilic is also hydrophobic. Absolutely correct answer. But don't make that mistake. Even hydrophilic messengers can achieve this long-term regulation, but they will need specific proteins, please. They can't do it by themselves. So see, you are connecting your signal transduction with the first. Yes, it's known as, for hydrophilic messengers, somebody has rightly said here, for hydrophilic messengers, epinephrine. So epinephrine increases which second messenger? Tell me, epinephrine. Somebody has put that in the chat, so I'll just explain him and everyone too. So epinephrine increases which messenger inside the cell? Cyclic AMP. So someone has rightly written there that epinephrine, if it wants to work at the level of gene, it will require cyclic AMP responsive element binding protein, the CREB that you have written. So it's not a crack, CREP, it's CREB, a cyclic AMP responsive element binding protein. It is B, not P, right? Don't dump it. So that is why understand the concepts that may be interrelated. Also, understand short-term regulation more, because short-term regulation is quick and more common, right? And I have enumerated differences. Every enzyme will undergo under specific short-term regulation. It's not a blanket treatment for all, except reversible covalent modification, and not even allosteric regulation, because allosteric regulation needs specific allosteric enzymes to be acted. Except reversible covalent modification, every other short-term regulation is very specific. So you can keep it as a blanket treatment for all enzymes and mess them up. For example, I told you substrate concentration is glucokinase. Will glucokinase ever be active in fasting or when you wake up? When you wake up in the morning and in fasting, can glucokinase be active? No. Now, how will you get better grades? One, that you already did it here. You are right in the lecture number 1, and you identified glucokinase cannot be active when I wake up. Yes, it will be inactive, or when I'm in fasting. Then you finish everything about glucokinase. Enumerate the difference between glucokinase and hexokinase. So the more and more you keep integrating and repeating, better grades. The more and more you are just stuck to, okay, I know glucokinase is affected by substrate concentration, which is glucose, and I'll get good grades. No. You finish everything about glucokinase in the lecture. Now you do comparison between glucokinase and hexokinase. What will that help you? That will integrate your overview with enzyme kinetics. Does that make sense? More you beat around the focus, better grades. Does that make sense? Product inhibition is by which enzyme? Anyone knows the enzyme? Product inhibition is by which enzyme? Anyone can recall the enzyme, which is predominantly under product inhibition? Hexokinase. Then do the difference between glucokinase and hexokinase. This is also very specific, conversion of proenzyme into enzyme. This is for pancreatic proteases. They cannot be active inside the cell. They have to be made active only in the lumen. So they are formed as proenzyme. This is common to all. Yes? Anyone has question? Anyone has any question? Are you following my pace? Is everyone good with my pace? Yeah, it's for you to note down, right? I'm not teaching. So reversible covalent modification is for blanket for all. You must know, don't mess that up. Insulin always and always dephosphorylates the key enzyme by using phosphatase. When you come to me in the next week, then I will tell you that is the dephosphorylated form active or inactive. For example, do you think insulin supports glycolysis? Do you think insulin supports glycolysis? Do you think insulin supports glycolysis? Yes. So if insulin supports glycolysis and you know insulin always and always dephosphorylates the key enzyme, which form glycolytic key enzyme will be active, phosphorylated or dephosphorylated? Which form they will be active? Dephosphorylated. That's all you need to do. First, you don't shake that concept that insulin always dephosphorylates the key enzyme. Then do not memorize which enzyme will be active in dephosphorylated form. Any enzyme that or pathway insulin supports, those rate-limiting enzyme will be active in dephosphorylated form. Can you mute yourself, please, unless you're asking question? Please. Do you have any question? Thank you. Allosteric regulation. Allosteric regulation is also tested big time. Why? Because allosteric, first of all, kinetics. Allosteric, that's why I think I'm not just going to tell you what is important for this lecture, because this exam is more about integration also. I will also connect other lectures. So here allosteric regulation is a short-term regulation, but don't forget to do allosteric inhibition along with this. You know also the criteria. What are allosteric enzyme? They have multiple subunit. Allosteric effector binds to the same site or other than substrate binding site. Allosteric effector binds to the same site or the other substrate binding site. Does the modifier when bind to the enzyme changes the conformation of the enzyme? Yes. That is why that smooth coordinated change is reflected in the form of what shape? What is the shape of allosteric enzyme when you draw the variables velocity versus substrate concentration? S-type, sigmoid, S-shaped. Then you can divide it into K-type, V-type. We will do that. But now allosteric modifiers, can it be positive, negative, both? Absolutely, either one of them. So for example, what is the purpose of glycolysis, do you think, in your body? Or what is the purpose of oxidative phosphorylation or ETC in your body? What does it give you? ATP. So tell me, will ATP, when I say ATP, when I'm asking you ATP as a regulator, I'm assuming it's high unless ratio is given. Please be mindful of the ratio. It's not always ATP to ADP. The whole concept changes if the ratio given is ADP to ATP. But I'm telling you that if the ratio of ATP to ADP is high, what will happen to glycolysis? Oxidative phosphorylation or ETC, will they be active or slow down? Slow down. So here, very, very important. You must know the primary allosteric modifier for every. Please mute yourself. Guys, please, unless you're asking question, please, because it disturbs my thought. So again, what we are talking about is allosteric regulation. You must know that you must know the primary allosteric modifier for a pathway, primary hormone for the pathway. For example, you just said that. So who is the primary turn off for glycolysis? What would you say? Which allosteric modifier is the primary turn off for glycolysis? High levels of ATP, glucagon later. I'm talking about allosteric modifier. Now, somebody who answers glucagon, see, this is how you affect your own grades. Question is not asking you, and you are picking wrong answer. I'm asking you, who is the allosteric, who is the primary allosteric modifier, I should say, though negative modifier, or what will slow down glycolysis, then you should say ATP, not hormone, because insulin, glucagon, they are not allosteric modifier. What do you mean by allosteric modifier? ATP, ADP, metabolites. Does that make sense? So insulin, glucagon, anything would not be the right answer. But definitely someone has answered. So I also clarify. Yes, insulin, glucagon work antagonist. So insulin stimulates glycolysis, glucagon may not. But still, please, the primary modifier of insulin is ATP levels, because when you come to me in the next week, please chart out what is the primary purpose of the pathway. That is the primary modifier. And when I ask what will slow down glycolysis, allosteric modifier, it should be high levels of ATP. Since the purpose of glycolysis is ATP, I would see primary effect is of ATP than just insulin on glycolysis. Does that make sense? Primary must be in your head. So this is how we will connect. So, but if you don't understand what is allosteric regulation, you cannot make high order connections in the next week. You know that proenzymes should not be active in the cells. If they are, they cause pancreatitis. Now, this slide doesn't mean pancreatitis is on the test. This slide means you have to bring in your drugs and clinical application, please. You can keep them isolated in medicine. Do that for me. What does insulin do always and always? Phosphorylates or dephosphorylates the key enzyme? Dephosphorylates the key enzyme, and this is not allosteric modification. Just because insulin, because this process is using ATP, this is not allosteric modification. What is this modification specifically? What is this modification? Phosphorylating, reversible covalent modification. Be mindful. Covalent doesn't mean irreversible. Covalent only means strong, right? Strong bonding. And then what kinase is this? See, you will understand signal transduction here. Epinephrine and glucagon in the last use which kinase to phosphorylate the key enzyme? Absolutely wonderful. Protein kinase A. So should you know protein kinase A, B, C, and G? Absolutely all of them. Which ligand is using it? So see here, you go further. If you just learn glucagon and epinephrine work through pKa, you pass. But if you learn what are the other pKas, B, C, and G, and who uses them, you get better and better grades. I'm giving you focus. The more you beat around that focus and know, you will get better and better grades. My aim is you all should pass. I'm giving you the focus. The more you work hard on that focus, you will give me better and better grades. That's the key for any test in medicine, right? You have to combine hard work with smart work, believe me. That's the only difference. Otherwise, everybody is working hard. Yes? So enzyme kinetics, stick to the learning objectives, understand the concept that inhibitor binds to the enzyme, not the substrate. Now you keep doing what will be the change in kinetics of the enzyme when a competitive inhibitor binds to the enzyme or a non-competitive inhibitor binds to the enzyme. So not just graph, the concept how competitive and non-competitive inhibitor works, but please see, you have to know normal to know abnormal. Unless you know that inhibitor binds to the enzyme, not to the substrate, you can't make that connection. Then, did I miss anything? No. Then I'm just giving you focus. The slide doesn't mean that the whole slide is on the test. I'm giving you a focus. Concentrate on that focus on that slide. Please don't forget to bring in, rather than the classification, no, this is memorization. Bring in which vitamins are which coenzyme. So when I come in the next lecture, next week lectures, you can absolutely connect. You don't need to draw this. You should be familiar with graph. Absolutely. This is not enzyme kinetic graph. You should carefully see first, whenever a graph is on the test, what are the variables on the x-axis and the y-axis. This is not kinetic graph. Then You should, yes, they are all same. A, B, C are same. So what is different? D. So no D first. A is the activation energy in the absence of enzyme. B is the activation energy in the presence of enzyme. C is the activation energy of the reverse reaction. What's only difference is D. So concentrate on D. D is constant in this graph. So that means do enzyme. What is D? Anyone? What's D? So the concept. What is D? D is known as Gibbs free energy. Now, don't forget to connect it with the electron transport chain. It is the difference in the energy of product to that of reactants. Is it altered by the enzyme? No, it's not altered by the enzyme. Yes. So some are absolute no, some are very specific. Concentrate on them rather than drawing these graphs and feeling happy about them. Only and only pathway I want you to know in and out as an outline that will really help you to answer a lot of questions or interconnecting questions is just glycolysis and TCA. Others, I will tell you what you need to know. Now, you don't keep memorizing the classification, waste of time, but you must know specifically this oxidoreductase because this is the major class doing oxidation in your body. And you know oxidation is always accompanied with reduction in your body, but this is the major enzyme and subenzyme dehydrogenase. So should you keep an eye on your oxidoreductase and dehydrogenase everywhere? Absolutely. Absolutely. You should know all your dehydrogenases. I will tell you especially which are for the rate limiting step coming up too. We have not learned so far, but oxidoreductase and dehydrogenase class you should be familiar with. Should you be familiar with clinical enzyme? Absolutely. Absolutely. You should know which are hepatic, which are cardiac, right? Do we do CKMB as cardiac? TROP-T, TROP-I, cardiac markers? Yes. Are they specific too? Are these enzymes specific too? So know their specificity, that's all. This is my enzyme, what it can, what can I as a physician order when I see this damage? For example, if you see a cardiac damage, will you order ALT? Will you order ALT? Primarily, no, you will order CKMB, TROP-T, TROP-I. So only the, especially the common, please. The common enzymes used and what tissue damage you would like to see them, know them. We have not taught you that rays of alanine transaminase and bilirubin. This indicates hepatic jaundice. This indicates hepatic jaundice. We have not taught you bilirubin, but we have addressed alanine transaminase here, right? So keep an eye on the more commonly used. Why there's mushroom here just to wake you up? Why is there a mushroom figure here? Have you heard about mushroom poisoning? Yes, it affects it affects hepatic cells and causes hepatic damage. So that's why this is more in alignment with this picture, mushroom poisoning. Well, you're not doing where it is used, but you should at least know tissue specificity for these enzymes, right? CKMB is for heart, LDH1 is for heart. Yes, absolutely. But don't go and Google mushrooms now, Sidhu, right? Yes, mushroom poisoning. CKMB is cardiac, so specificity only. LDH1 is cardiac. LDH1 is cardiac. So specificity. TROP-T, TROP-I are cardiac. Now, some people do kinetics, some do graph. I don't want you to stop here. You must understand what is the initial velocity, initial part of the reaction. No product inhibition then. Vmax is what? Tell me. What do you mean by Vmax? Enzyme has reached? Vmax enzyme has reached what? Vmax reflects efficiency, but what does Vmax show? That the enzyme has reached its saturation. Vmax can be altered by a substrate concentration, yes or no? Can Vmax be altered by substrate concentration? No, Vmax is now entirely dependent on the enzyme, because when we say saturated, what is saturated? The enzyme has been saturated. Their active sites have been saturated. Then you should, yes, somebody said, which is also very important, Vmax reflects efficiency of the enzyme. Km reflects the affinity of the enzyme. You know, when you put a bracket here, this is substrate concentration. Some do by kinetics, but I encourage you to do by kinetic equation, which is VO is Vmax times S upon Km plus S. But I will encourage you to use graphs to understand any concept. I'll just explain you few. I hope this is helping. I hope this kind of review is helping. Long term, yes, very good. Somebody said long term regulation. Can it change Vmax? Absolutely. Very good interpretation and integration. This is what I want you to do. Long term regulation alters Vmax, because Vmax is dependent on number of enzyme. Induction and repression is doing what? Inducing the enzyme and its concentration, or repressing the enzyme and the concentration. So absolutely, Vmax going up and down is not just, sorry, it's going down is what? Competitive or non-competitive inhibition? Vmax going down. Is it competitive? It's non-competitive inhibition. But somebody had a very good point too. Apart from Vmax going down being non-competitive, it is also repression, which is a part of long-term regulation. Repression is also reflected same as in the graph I'll show you as non-competitive inhibition. Somebody rightly pointed that. Remember, you will not understand that unless you know Vmax can be only altered with the help of enzyme. Right? Then first order and zero order kinetics. First order means proportional flow. Zero order means constant flow. Now don't stop here. Extrapolate to your transporters. Which transporters follow this initial part of reaction, which is proportional flow? That means glucose only flows in them when it's high after meals. Yes, GLUT2. Don't stop then too. GLUT2 site. Where is GLUT2? Zero and first order. Do we need to know this? You need to know what is zero and first order, not by drawing equation, knowing that this first order is proportional and zero order is when there is saturation constant. Yes, just this graph, how I have explained, don't draw any equation. This is first order and here is zero order where you reach saturation. But not just that, integrating it with glucose transporters. First order, who follows first order kinetics? GLUT2. Where are they? Liver and pancreas. Why they are there? Because liver and pancreas want proportional flow. They only want glucose to be going in the cells when glucose is high after meal. One more thing, somebody was confused with lumen. Please, lumen is only in the intestine. There's no liver lumen, pancreas lumen. They don't have lumen. Lumen, you talk in intestine. So when I'm saying lumen, I'm talking about intestine. When I'm saying liver and pancreas, I'll talk about cells. So very good. Somebody said, give what would be an example? I will tell you what is the example. Very good. Somebody answered that. So first, let's one step at a time. First order kinetics is extrapolated by GLUT2, where are GLUT2, liver and pancreas. Why they need GLUT2 transporters? Because you don't want glucose to flow in all the time in liver and pancreas. You want them to flow in only when glucose is high after meals. Because if you constantly flow glucose in liver and pancreas, liver is the chief site of metabolism, will be exhausted. Pancreas is a chief site of insulin release. There will always be insulin release. You cannot do that. So GLUT2 have a proportional flow. Who has the constant flow? Somebody very well answered here. I'm glad you're answering questions. If you're answering questions when I'm saying you're half prepared, all you need to do is go back, finish these lectures right in this weekend, right? Glut, who follows zero order kinetics, which is independent of substrate concentration, is, yes, glut 1 and 3. Where are they? Brain and RBC. Why do you want a constant kinetic glut 1 and 3 there? Because RBC is entirely dependent on glucose and brain is primarily dependent on glucose. Yes, they would die otherwise. You cannot afford a proportional kinetics in 1 and 3. So you should know all the transporters, absolutely. Their kinetics, absolutely, right? And when you RBCs, yes, mature RBC lack mitochondria, but that distinction is not helping glut transporters. In a way, it is helping because RBC cannot use any other fuel other than glucose. So they are entirely dependent on glucose, Amanda. That's a good point too. Brain, I would say 1 and 3 for brain and RBC. Not wrong in saying that. If the syllabus has specified it, why not pay attention on that? Right? Glut 1 and 3, brain and RBC. If there's a distinction, make it. That's okay. But the important thing is they both have a constant kinetics. Does that make sense? And reason being, brain primarily dependent on glucose, RBC entirely dependent on glucose. Did I answer you, Andrew?
Oh, yeah, thank you. It
Yes.
was just a memory trick. Thank you.
Yes, no worries. And also don't forget to know the double reciprocal part. Both can be tested, but this is a straight line extrapolated back. And why you need to understand the x-intercept and y-intercept is just, I will tell you, for graphs of competitive versus non-competitive inhibition. Km is not reflecting efficiency, it's reflecting affinity of the enzyme for substrate. This is first order kinetics in the initial part of the reaction. This is zero order kinetics. Someone had a very beautiful concept. I like students answering in my interactive session because that brings my thought to is of repression and induction. So again, this is first order. I have too many I have chat open and everything open. Give me a second, please. Again, this is first order kinetic. This is zero order kinetic. Vmax is not dependent on the substrate. It depends on the enzyme concentration. When Vmax is reached, means saturation is reached. Extrapolation or everything is given here. But right shift is altering what? Km or Vmax? Right shift of this curve, for example, I ask you, is altering Km. Is Km increasing when you are shifting right? Yes, is Km increasing when you're shifting right? So where does Km increase? Competitive or non-competitive inhibition? Km increases in competitive inhibition. Absolutely. Right shift is inhibition, left shift is activation. Vmax moving up is activation, Vmax moving down is inhibition. Now, one more concept that a student has brought in, which is absolutely wonderful, is induction and repression. Also the same as moving Vmax up and down. Yes, absolutely. I'll show you that, right? Some people understand by kinetics, some are by graph. I will encourage you to use graphs. This is first order, then this is zero order kinetics. Just a second, too many. This is first order and this is zero order kinetics. You can do it by equation 2 if you like that. If S is much larger than Km, see, this is the same interpretation. When S is much larger than Km, do you reach saturation? Does the reaction reach saturation? Because you can neglect the Km here in the denominator. So either you do by equation or you do by graph. I would encourage you to use the graph and be familiar with them. Is high Km low affinity? Is high Km low affinity for enzyme for the substrate? So who has a higher Km, hexokinase or glucokinase? Who has a higher Km, hexokinase or glucokinase? Higher Km has? No glucokinase. Who has? Oh, yes. Sorry. Yes, glucokinase has a higher Km and higher Vmax. Why is it like that? Glucokinase is with liver pancreas? Absolutely. Glucokinase has a high Km. So why is it like that? Why do liver and pancreas have glucokinase, not hexokinase? Because high Vmax means enzyme of good efficiency or affinity. Yes, now that, but I'm not asking blood transporter. So see, there you have to, again, when a question is asked specifically, if you stick to that, you would answer them better. So I'm not asking you blood transporter. I'm asking you why glucokinase is in liver and pancreas. And your answer is right, but it's not pertaining to my question. Your answer is absolutely right. There are blood transporters on liver and pancreas, they do proportional flow, but that has nothing to do with glucokinase. One thing it has to do with glucokinase, that glucokinase is also active, and what is high? When is glucokinase active? What is high in the circulation? Glucose. So GLUT2 help glucokinase. When glucose is high after meal, GLUT2 is only helping glucokinase to push the glucose inside the cell. But glucokinase has to be highly efficient. So it is the efficiency of glucokinase that we need in liver or pancreas. That has nothing to do with GLUT transporter. Now, this actually mistake students usually make. They think GLUT transporter and glucokinase is the same. No. GLUT transporters are on the surface, pushing the glucose inside the cell. Glucokinase is inside the cell, turning on glucose oxidation, always the first enzyme. Phosphorylation glucose, only one of them is able to give time. Okay, it came to this revelation. Now, phosphorylation and dephosphorylation of glucose. Why would you do that? Where is it given? Questions at this question I'll take at the end, Diego. I don't understand what do you mean by phosphorylation and dephosphorylation of glucose. There's nothing like that. It is phosphorylation and dephosphorylation of the key enzyme. Glucose can be phosphorylated and there are enzymes. That's not regulation. I'll please take your question later when saying, okay, that's great. If you have something to memorize that. Okay, so you should know glucokinase, hexokinase. There is a reason you have enzyme of high efficiency in liver and pancreas, because the liver is the chief site of metabolism, pancreas is to secrete insulin. Understand with more concepts in biochemistry, you will do better. If you do more memorization, might not do that great. Does that make sense? Ask yourself a relevant why. Secondly, I want to tell you is please don't be confused. Look at this here. Glucose transporters are right at the top on the surface, and glucokinase is inside. 1 and 3 is at the top. Hexokinase is inside. They are not related. Also, this is a very common mistake students make, especially in the coming week, when they ask you around a step. For example, if I ask you glucokinase is deficient, tell me what will accumulate in the cells. If glucokinase step is deficient, what will accumulate in the cells? Glucose. And what will be low in the cells? What will be low in the glucose 6-phosphate? First, concentrate around the step if they ask you any enzyme defect. Then if you don't get an answer here, you go up and down. For example, not now, later on I'll give. The common mistake students make, they keep going up, up, and up. No, first you concentrate and see if your answer you can get the answer in the choices from here. If you don't, then only you branch out. So that's why I'm telling you, if you want to do well in this exam, be very focused, be very focused. Focused on the step, focused on the rate limiting steps, focused on the primary hormone, primary metabolite, primary enzyme. Everything else is a background noise. And what do we do with background noise? Do we pay attention on that? Do we pay attention on background noises? Nope, just says I have to neglect the background noises when students don't mute themselves. Right? Does that make sense? So know these hexokinase, glucokinase very well. Know competitive and non-competitive inhibition. I just have this slide. Also, sorry, by knowing the concept, there's a structural similarity. There's a competition. You can overcome inhibition. All these facts. So tell me, in competitive inhibition, is there an ESI complex? Do you see an ESI complex? Ever? No. There is never an ESI complex in a competitive inhibition. Either the substrate will bind or the inhibitor will bind. But remember, what's constant is the enzyme. Then you do what is competitive inhibition? Graph? Absolutely. Absolutely. Get used to graph. Sometimes students are confused with the how to do it quick. So if it is a V by S, see what is changed. If it's 1 by V over 1 by S, see what is unchanged. Also, please know, if you want to understand concept, when Km is increasing, should it move towards positive or right side? This line should move right side if Km is increasing. Understand that, which is competitive inhibition. Now let's look at Vmax. If Vmax is decreasing, but this is 1 upon Vmax, where will be the line move, up or down? In non-competitive inhibition, Vmax decreases. So where will the line move? Up. That is why always and always control inhibitor line is above the control line. It's above the control line with increasing inhibition. So be mindful of these two graphs, absolutely, but also concept. Will you see ESI complex in non-competitive inhibition? Absolutely. Are both reversible? Absolutely. Is covalent modification reversible? Absolutely. Allosteric? Absolutely. So when do you call it irreversible? When binding is covalent. When you call it suicidal? When binding is not just covalent, it also, inhibitor also kills the enzyme. Right? So who will tell me what happens to the number of enzymes when a non-competitive inhibitor binds to the enzyme? Are the number of enzymes reduced? What happens? Yes. So when enzymes are reduced, which parameter changes? Vmax or Km? Vmax. So also pay attention on the differences, because otherwise you will memorize this, can memorize this. Again, look at this. Is this non-competitive inhibition? How do you say it's non-competitive inhibition? First of all, inhibitor line is always above the control line. But what tells you on the right side it is non-competitive inhibition? Which intercept is unchanged? Yes, x-intercept remains unchanged. So you quickly know this is non-competitive. This can't be competitive. Does that make sense? See, this is the concept. You can practice this when you have done everything. This is somebody pointed out. Absolutely. These two go together. Vmax decrease is not just non-competitive inhibition, is also repression. This is a high level of understanding if you can connect that repression is also Vmax going down, though Vmax going down is non-competitive inhibition. So the concept, what is activation, what is inhibition? Examples always help. Is aspirin everywhere? Aspirin is everywhere, right? We get headaches all the time, right? Aspirin, you should know aspirin. Aspirin here is just an irreversible inhibitor. Aspirin, cyanide, nerve gas poisons, they are just irreversible inhibitor. So know the graphs well. You have any confusion in the graph? This is just to wake you up, right? This is not, uncompetitive inhibition is not being tested, please, on the test. We have never taught you. So just to wake you up. Right? Now, what is happening to the Km line? Is Km line moving right or left? What's happening to the Km line? Is it moving right or left? Left. Very good. What's happening to the Vmax? Is it moving up or down? Vmax is moving up. Now you tell me, since Km is moving left and Vmax is moving up, what is the outcome? Are both Km and Vmax decreased or no? What does this graph show? Are both Km and Vmax decreased or increased? Both decrease. Absolutely. Km is decreased and Vmax is decreased. So it's the interpretation. How can you understand these graphs? Any way you can understand them, do it. You don't really have to do this uncompetitive graph, but it will help you know that your concepts are correct, right? So, you know, short-term regulation. One thing I want to bring this, which is also very, very important, and I think, Amanda, you brought that up, is compartmentalization. You must know which steps are cytosolic, which are mitochondrial. Absolutely. You must understand whenever mitochondria is not working, for example, physiologically, do RBC lack mitochondria? Yes. In cancers, are mitochondria affected? Yes. In both cases, or LHON, MERRF, MILAS, wherever mitochondria is affected, tell me which processes will go up when mitochondria is affected. When mitochondria is affected, which processes will go up in your body? For example, in cancer, in mature RBC, cytosolic. Just say cytosolic, not just glycolysis, because glycolysis is aerobic and anaerobic. Just say P53. I'm not talking about cancer here. I'm saying whenever mitochondrial disorders, sorry, whenever mitochondrial processes are not working in your body to compensate, to keep your body alive, what kicks in? Cytosolic process. You must know which process is cytosolic. For example, is oxidative phosphorylation cytosolic? Is oxidative phosphorylation cytosolic? Yes or no? No. So can it happen in the mature RBC? Can it happen in the mature RBC? Can oxidative phosphorylation happen in the mature RBC? No, because mature RBC need mitochondria. So you understand my point? You should understand which pathways or processes are cytosolic and mitochondrial. That will also help you, which is very, very important, to understand oxidative versus substrate level phosphorylation. So whenever mitochondria is missing, cytosolic processes push up. And even substrate level phosphorylation pushes up because oxidative cannot happen. So you should understand mitochondrial versus cytosolic, oxidative phosphorylation versus substrate level phosphorylation. Does that make sense? Please pay close attention on both of them, right? Let's go further. Yes, don't forget Vmax moving up and down is induction and repression respectively. You know allostric to identify first by variables. This is velocity versus substrate and shape is S-shaped. It has to be allostric enzyme. They do not have a KM. There are characteristic of allostric enzyme. Now, who will tell me in signal transduction which enzyme is allostric in signal transduction? Which enzyme is allosteric? There's an enzyme which is allostric in signal transduction. Has anyone? Enzyme I'm asking, and you're telling me insulin. Insulin is a hormone, please. I'm asking you enzyme, which is IRSR, insulin receptor substrates. Anyone who knows that one enzyme of signal transduction is allostric. I will tell you oxidative versus substrate level phosphorylation in the next week. Is somebody has a good question? Let me answer that. Sorry. First, tell me what is which enzyme, so I don't lose that. Which enzyme? Protein kinase A. Protein kinase A is an allostric enzyme, and its allostric effector is very good. Somebody said that is cyclic AMP. It's a very good, very good. It's a cyclic AMP dependent protein kinase A. But somebody has a question. What's the difference between substrate level phosphorylation and no? There are no cytosol. No. So let me go back again. There is nothing called cytosolic phosphorylation. There are two things. First of all, you should know the difference between mitochondrial and cytosolic process. Okay. Secondly, you should know the difference between oxidative phosphorylation and substrate level phosphorylation. Am I clear first, Rumen? So there are two concepts you need to know. Mitochondrial versus cytosolic process. Secondly, you should know the difference between oxidative and substrate level phosphorylation. What is oxidative phosphorylation? I will show you in the subsequent slide. But substrate level phosphorylation means the energy is so high. First of all, what is oxidative phosphorylation? Good question, though I wanted to talk in the subsequent slide, but I'll clarify here. Oxidative phosphorylation means you synthesize ATP with the help of ETC. Substrate level phosphorylation means you synthesize ATP without ETC. How does that happen? Sometimes substrate itself has so much energy that it can release ATP. So that is the difference between oxidative phosphorylation and substrate level phosphorylation. Oxidative phosphorylation means you need ETC to form ATP. Did I clear that, Rumen? In substrate level phosphorylation, you don't need ETC to synthesize ATP. That's why you should know when oxidative phosphorylation can happen, when substrate level phosphorylation can happen, when mitochondrial steps can happen, when cytosolic steps can happen. ETC because of the oxygen acceptor. What do you mean by ETC about the oxygen, please?
So you mentioned that it's ETC that synthesizes like the ATP for oxidative phosphorylation. Is that because of the oxygen that's being accepted towards the end, or no, that's not connected?
No.
That what you mean?
No, I will tell you that could be, that is why, because that is, we call it oxidative phosphorylation, not for that reason. You call oxidative phosphorylation because you first oxidize the substrate, right?
Oh, here we
And
go.
then, yes.
Like,
So the oxidation of substrate is linked with ETC to form phosphate. That is why they together are known as, that is why they are coupled. They are known as oxidative phosphorylation. But yes, there's nothing wrong in saying that since you need oxygen for oxidative phosphorylation, you can say that, but that's not the primary reason. Not wrong. Not wrong to say that. Diego, I mean to say that she's asking, why do you call it oxidative phosphorylation? You call it oxidative phosphorylation because you're coupling substrate oxidation with ATP production in ETC. That is why you call it oxidative phosphorylation. Not just because oxygen is used in electron transport chain. Did I clear that? Got it. When you said insulin support value, do you mean it supports regulation through pKa? Very good question. Very good question. So when I'm saying insulin supports glycolysis, which your first statement is right, will insulin support glycolysis through pKa or phosphatase? Anyone? When I'm saying insulin supports glycolysis, will insulin support glycolysis through activating pKa or phosphatase? So your second statement is wrong. The first statement is correct, that insulin supports glycolysis, but not through pKa, but phosphatase. Yes, because phosphatase dephosphorylates the key enzyme. No. How can it inhibits through phosphatase? Okay, now I think we are going too far in the second week, but I'll explain you this one time. So we stick to this week, okay? We stick to this week. Insulin always dephosphorylates the key enzyme, right? Whether this enzyme should be active or not depends on the pathway. So I just gave you an example that insulin supports which pathway? Glycolysis. So you tell me which form of glycolytic rate-limiting enzyme be active in phosphorylated form or dephosphorylated form. So that doesn't change. The first statement that insulin dephosphorylate the key enzyme by activating phosphatase is universal. It's not variable, please. It is not dependent. Okay? No. Enzymes are not active and they are dephosphorylated. Okay. Listen to me again and let's talk about this next week. Enzyme insulin through phosphatase always dephosphorylates the key enzyme. Just let's stick to that. That doesn't mean that the dephosphorylated form of every enzyme is active. It depends. If insulin supports that pathway on the enzyme, that enzyme would be active in dephosphorylated form. No, it will not be. Please, let's not go to hexokinase, glucokinase next week. I will tell you how hexokinase is, first of all, hexokinase is not subjected to covalent modification. So you are not doing phosphorylation and dephosphorylation there. Only specific enzymes are subjected to covalent modification. So that would be a wrong even question to frame, is hexokinase phosphorylated? Hexokinase does not undergo predominantly covalent modification. Does that make sense? Let's talk about that in the next week. Everybody, can you just stick to signal transduction, insulin, phosphatase, dephosphorylation? Is everybody good with that? We are confusing the students, please. Does that make sense? Let's not talk about any other enzyme right now. You have not learned the pathway. So allostric, know the characteristic, because I want you to stick to this week, please. We haven't learned anything yet, okay? So, but insulin, phosphatase, dephosphorylate, don't mess that up, please. We will see later on which is active or inactive. Don't make any statement, please. So allostric enzyme also binds to site other than substrate binding site. Only thing what they do is induce conformational change. They do not have a KM. They are only K and V type, please. Okay. This is only concept. You should not draw do calculations. There are no calculations on the test. All you need to know concept. Vmax by 2 is Km, but Km, be mindful, is substrate concentration. Then next question is to understand first order versus zero order reaction. So I will only do one, which is zero order reaction. So let's see zero order. What does zero order means? S is much larger than Km, right? When you need to reach saturation. So if you compare the substrate concentration of A with Km, S is larger than Km for A. So that means this is reaction is at Vmax. So your answer should be 20. Again, the concept that when S is much larger than Km, reaction reaches saturation and reaches Vmax. Is Vmax altered by substrate concentration? No. Is Vmax altered by enzyme? Absolutely yes. Please stick to the learning objectives. First, make a connection between oxidation versus reduction. Know delta G, which is difference in product to that of reactants, in 3 ways. When delta G is negative, is reaction spontaneous? When delta G is negative, is reaction spontaneous? Also, it is exergonic. Okay, spontaneous goes to completion and exergonic. So you must understand, first of all, delta G negative means spontaneous, exergonic, going to completion. And oxidation reactions are having delta G negative. Thirdly, you should know if delta G is negative on of high magnitude or quantity, it is irreversible. That step becomes irreversible. So all the irreversible steps have delta G negative. Is it safe to say that? Is it safe to make that statement that all the irreversible steps have delta G negative? Absolutely. Only when delta G is negative and of high magnitude, then the step becomes irreversible. Right side difference in product to that of reactants is positive. It doesn't go to completion. Right side steps are reduction. Sorry. Yes, somebody has a question. So right side, yes, right side is endergonic, needs energy. Most of the reaction, unfortunately, in your body are endergonic, but they go to completion by being coupled with exergonic reactions. And most common exergonic reaction is, sorry about that, ATP. ATP hydrolysis is the most common exergonic reaction coupled with endergonic reaction. All irreversible, yes, all irreversible reactions, you're right, has delta G negative. Yes. You're right, absolutely right. Now you'd go ETC. First of all, you should understand oxidations are exergonic. They are accompanied by release of energy. But if you release all that heat, that is uncoupler. We don't want to do that. So what does body do? The catabolism is mainly oxidation, accompanied with reduction, mainly oxidation. So should you know the enzyme oxidoreductase, subclass dehydrogenase? Very well. Should you know the vitamins involved? Very well. But oxidation in our body, which is loss of hydrogen, and the exergonic energy release is slowly and very coordinated way trapped in the form of ATP. almost 90% of energy is trapped. So if ATP synthesis, if this energy is not trapped as ATP, then the body will go into hyperthermia, which happens in uncoupler. You must understand well that concept. You must understand uncoupler and its hyperthermic effect, right? And which drug toxicity is also uncoupler? Any drug toxicity? Any drug toxicity or drug in high doses? Aspirin. Aspirin in high doses is also an uncoupler, right? So first, normal. Oxidation, exergonic, release energy, trapped. How does oxidation happen? Loss of hydrogen. They carry all the energy. The energy is known as Gibbs free energy and trapped as ATP. NADPH is not a marker of ATP. Do not choose NADPH as ATP indicator. Indicator of ATP is either ATP or NADH and FADH2. You should very well know this slide with respect to oxidative versus substrate level phosphorylation. You know substrates are oxidized and who's getting reduced? When substrates are getting oxidized, who gets reduced? There's always oxidation accompanied with reduction. When substrates are oxidized, what is getting reduced? Anyone? What is getting reduced is the coenzymes, NAD more than FAD. You have to regenerate them and form ATP. So that is why this is oxidation coupled with phosphorylation. This is oxidative phosphorylation when you need ETC to produce ATP. When you don't need ETC to produce ATP, that is substrate level phosphorylation. Please know this very well. If your ETC is not functional, your mitochondria is not functional, or you don't have oxygen, you cannot do oxidative phosphorylation. Then what will you turn on in body? When oxidative phosphorylation is not happening, what will be turned on in the body? If there's no oxidative phosphorylation, either cytosolic process or substrate level phosphorylation. Substrate level phosphorylation, both will be turned on. Either so when there is no ETC, mitochondria, oxygen, you cannot do oxidative phosphorylation. So what will turn on in your body? Either cytosolic process or substrate level phosphorylation, but both are different. Know your vitamins. Redox potential, you will not be asked to calculate. All you need to understand is that one substrate is oxidized and the other is reduced. This keeps on going until all the H plus and electron need oxygen. That is all you want to do, a smooth coordinated flow of H plus and electron to final oxygen. Always know that high reduced coenzymes in the matrix shows good ATP. So tell me, this is what we are going to do, citric acid cycle, TCA cycle. They are all going to give you all these reduced coenzyme. But you must understand that if I have a niacin deficiency, then complex 2 will not be inhibited. So you have to make that application then. If they are testing you on niacin deficiency, complex 2 step will not be affected. What will be affected is complex 1. So keep an eye on your vitamins, coenzyme. I do want to bring these shuttles though, because when you oxidize your food stuff, please know your chief. I'll go back. When you oxidize your food stuff, your main metabolism is TCA, which is coming up next. And when you oxidize food stuff, what do you get? A lot of reduce coenzymes. But this is in the TCA cycle is mitochondrial, which is the main pathway for oxidation. So most of your reduced coenzyme are where? Actually, because TCA is mitochondrial. When you do oxidation, where are your most of the reduced coenzymes? Where are they? Are they in mitochondrial matrix? So when you do good oxidation, no, not in inner mitochondrial membrane. Yes, it is in the mitochondrial matrix. That is proton flow. Proton flow is from matrix to intermembrane space back to matrix. But no, the reduced coenzyme, they are impermeable to inner membrane. They cannot be in inner membrane. When you do good oxidation, and that means you have collected a lot of reduced coenzyme, which are indicator of ATP, they are in the matrix. But tell me, how will you bring the reduced coenzymes that are outside the matrix? How can they come in? Because electron transport chain is in the matrix. You understand my point? Where do shuttle come in, which we will introduce? Then you use shuttle. Shuttle means, so most of the oxidation is by TCA cycle. With oxidation, you form a lot of reduced coenzyme. So most of the reduced coenzyme are in the mitochondrial matrix. But how will you bring those reduced coenzyme which are produced outside the mitochondria? For that you need shuttles. Because you need shuttle to push those reduced coenzymes, hydrogen and electron in the matrix, because electron transport chain is in the matrix. So understand the shuttle when I'll talk about TCA cycle. Shuttle is like take you from one place to the other, right? Yeah, we will do that. We will do the shuttles, not now, TCA cycle, next week. You know all the five complexes. Fifth complex is also known as ATP synthase complex. Know the flow of H plus an electron. I have explained you well with this slide. There's always unidirectional, not back and forth. Yes. Also understand the function of the complexes. Also understand that CoQ is oxidized, who gets reduced? When CoQ is oxidized, who gets reduced? When CoQ is oxidized, who gets reduced? Anyone? When CoQ is oxidized, who gets reduced? Cytochrome C. When cytochrome C is oxidized, who gets reduced? Oxygen. First, know this normal. Understand substrate linked to FAD, bypass complex 1. Understand that when you inhibit complex 4, can oxygen be reduced? So please understand those concepts too. When you inhibit complex 4, can oxygen be reduced? No. So do all that, then just memorizing the inhibitors. Definitely first memorize the inhibitors of 1, 2, 3, 4, but understand which function can be deranged if you have an inhibitor, right? Also, please understand that our main aim is all the H plus and electrons finally go to oxygen. Reactive oxygen species are always produced with normal metabolism. We'll talk about that in subsequent lectures. So I'm just bringing this up. You might have reactive oxygen species in the next week, but how do we deal with them? What is the difference between oxidative and substrate level phosphorylation? Are you clear now? Yes? Everybody? Yes? Are you clear with oxidative versus substrate level phosphorylation? Everyone? Yes. Where are most of your reduced coenzyme? In the matrix. If anything is outside the matrix, they will be brought in by shuttle. We'll talk about that shuttle. But know your inhibitors of complexes versus ATP synthase complex versus uncouplers. Uncouplers, what do? They dissociate oxidation from phosphorylation. So you must know the effect of inhibitors on ETC function, oxygen consumption, and ATP synthesis. I will explain you that. These three, please know on all these three. So give me an example of complex 2 inhibitor. Any example of complex 2 inhibitor? Complex 2, anyone? Malonate. Antimycin is 3, malonate. Substrate linked to FAD, bypass complex 1. Who is the chief controller of ETC? ATP. High ATP will do what to ETC? Tell me. What will high ATP do to ETC? What will high ATP do to ETC? It will slow down. You must know primary controller, hormone, enzyme of each process or pathway. Did we talk about aspirin as an uncoupler? Did we talk about aspirin in high doses as uncoupler? Right? Keep an eye on your drugs, please. I asked you this, inhibitor of complex 2, you said malonate. Know their effect on all these three. I will just explain you with this graph if you understand things well. You know the difference between carbon monoxide and cyanide poisoning. Where is oxygen used as treatment? Where is hyperbaric oxygen used as treatment? Carbon monoxide. Where binding of the toxin to the ETC is covalent. Where binding of the toxin to the ETC is covalent cyanide. What's the antidote in cyanide poisoning? What's the antidote in cyanide poisoning? Amyl nitrite, know its effect. You know the focus of cyanide versus carbon monoxide poisoning. Yes, absolutely do it, right? Thermogenin, 2,4-dinitrophenol, 2,4-dinitrocresol, aspirin are uncoupler. Aspirin is especially uncoupler in high doses, right? This is what somebody was asking me. Why do you call oxidative phosphorylation? Because you're coupling oxidation of foodstuff or etc with formation of ATP, and you need electron transport chain for it. So this is known as oxidative phosphorylation. Cyanide prefers amyl nitrite. Yes, only because you gave an antidote amyl nitrite during cyanide poisoning. Otherwise, cyanide will have affinity to Fe+3 of complex 4. But you're right. When amyl nitrite is pushed as an antidote, now cyanide will prefer amyl nitrite sorry, again. So you're very right that when amyl nitrite is given an antidote, cyanide will now prefer Fe+3 of methemoglobin than Fe+3 of complex 4. Does that make sense? Cyanide is not preferring amyl nitrite. Amyl nitrite is forming methemoglobin, and cyanide is now going towards Fe+3 of methemoglobin, then going towards Fe+3 of complex 4, right? Let's do this quickly. Please see there are 6 lectures. I'm trying to wind up. Just give me a few more minutes, right? Is everybody okay? Is everybody good? Is it helping? Yes, then I'll open for question and answer. Now look at this. You have mitochondria, you have substrate. What is this substrate linked to in electron transport chain? Complex 1 or 2? What is the substrate complex 2? So you have mitochondria, that means you have ETC, you have substrate. Now who will turn this process on? High ATP or high ADP? What is A? What will turn this process on? High ATP or high ADP? High ADP will turn this process on. And remember, when the process is on, this is oxygen concentration, not consumption. So will the oxygen concentration, this is an experiment, right? I just want you to put an eye on this to understand how you need to know concept, then memorize it. Oxygen concentration is falling in this graph. Does that mean oxygen consumption by ETC is increasing? Does that mean the oxygen consumption by ETC is increasing? Yes. So is this normal? Absolutely normal. This is absolutely normal phenomena. Healthy body. You have mitochondria, electron transport chain, all the enzymes, substrate, and ADP, high ADP. ETC turned on, function of oxidative phosphorylation turned on, oxygen concentration decreased, that means oxygen consumption increased. Absolutely fine. Now what happened here, somebody in the experiment added an inhibitor and oxygen concentration slows down. That means oxygen consumption by ETC is low. When does that happen? Does it happen in the presence of uncoupler or other inhibitors? Can B be uncoupler? Everything was going good, but B came in and there was a slow drop in oxygen concentration. That means the oxygen consumption by ETC is decreased. So can B be uncoupler? Yes or no? Can B be uncoupler? No, because uncoupler increase oxygen consumption by ETC. ETC does not know where to stop. So not B, but C is an uncoupler. C is an uncoupler because you see another steep fall in oxygen concentration or increase in oxygen consumption, which is abnormal. So A is healthy, B is anything other than uncoupler, C is uncoupler. Now you tell me, you see a fall in oxygen concentration after A, which is healthy. You see another fall in oxygen concentration or increase in oxygen consumption after adding C. Are they both same? Is A and C same? What's the difference, which is not shown in this slide? What's happening at the level of C? First of all, C is an uncoupler. There is increase in oxygen consumption. But how is A and C different? A is healthy and C is unhealthy. How are they unhealthy? How is C unhealthy? Because apart from increase in oxygen consumption, what is not being formed? Yes, so what is not happening here, which is happening on A? ATP is not being formed at C, and that is why all the energy is released as heat. Absolutely. Good to get graphs, make your flashcards. I don't discourage that, but understand concepts than pure memorization. So yes, C should be an uncoupler, but there should not be any ATP synthesis, right? Does that make sense? Concepts. I have some questions. They are on the review PowerPoint on Canvas too. I will share this slide too. Don't stress with signaling. First of all, no hydrophilic versus hydrophobic. Hydrophobic use intracellular messenger and work at the level of gene. With hydrophilic, I want you to know three hydrophilic signal transduction mechanism well. Heptahelical, growth factor, and insulin. Please know all of them well, these three. Rather than JAK-STAT and TGF beta, no. Does that make sense? Please, these three do well. Not just what not just what is in each of one of them. For example, when I say monomeric GRAS, where is it? Is it in growth factor? Is it in heptahelical? So I said you need to do three in details. First one is growth factor signaling. Second is heptahelical receptors. Third is insulin. Heptahelical, growth factor, insulin. Yes. Yes. Okay. Secondly, order is also important. I'll show you how. Steroid, thyroid, intracellular. Act at the level of gene. Hydrophilic, use plasma membrane receptors. I will tell you how you need to know everything. Are trimeric G protein same as monomeric G protein? Where is monomeric G protein used? GRAS. Which signaling is GRAS used? Which signaling is GRAS used? Anyone? GRAS insulin can also use it, but again, see, where insulin uses it so low, but you are answering insulin. You have to change that concept too. More common and primary first. When I say GRAS, growth factor should come to your mind, then insulin. Does that make sense? Does that make sense? That will also help you get good grades. When I'm saying GRAS, you should first say growth factor. Heptahelical receptors, trimeric G protein, GS, GI, GQ, all of them, right? I will tell you only by showing you one example how you should know them. You must know application. We are not teaching you this so you don't relate. You should know cholera versus pertussis. There are drugs too like sildenafil. Bring in drugs and clinical application. One thing students get very confused is, please know cholera affects GS, pertussis affects GI. But cholera by modification makes GS persistently active. Pertussis makes GI persistently inactive. They are both toxins not acting at the same site. Outcome is the same. Yes. So insulin, what has this to do with phospholipase C gamma? Because insulin also uses phospholipase C, just like epinephrine alpha-1 GQ, but instead of epinephrine using phospholipase C beta, insulin uses phospholipase C gamma. You don't need to know that, but I'm still answering your question. Phospholipase C gamma has to differentiate from epinephrine phospholipase C, which is beta. But I have enumerated it in the lecture. Did I answer you? How do toxins of GPR lead to the increase in cholera mix and pertussis free? So instead of, now know that instead of knowing GTP and GDP, the modification is not GTP and GDP. The modification is ADP. ADP modification by cholera is on GS, making it persistently active. However it is. Modification for pertussis is on GI and makes it persistently inactive. So don't get lost in GTP and GDP. The modification is ADP. For cholera, modification is on GS. For pertussis, it is on GI. Outcome is cholera makes GS persistently active. Pertussis makes GI persistently inactive. Does that make sense? In whatever normal state, it should be bound, GTP or GDP. That you do later. First, make that concept clear. Yes, how does it increase cyclic AMP? If cholera is persistently activating GS and pertussis is persistently inactivating GI, what should be the outcome? If GS is active or if GI is inactive, what second messenger will increase inside your cell? If GS is active, always, GI is inactive, what will increase inside your cell? CAMP. This is how it increases. Does that make sense? Yes. I can answer your question later. I want to tell a few more things so that I can wrap up, please. I will open the question and answer. But GS activation and GI inactivation have the same outcome. This is how they affect it. Also, please fill in the cascade. Epinephrine, beta, GS, adenylyl cyclase, cyclic AMP, protein kinase A. Bring all your protein kinase A, B, C, G. For example, who uses protein kinase C? Which ligand uses protein kinase C? If there's protein kinase C, what comes to your mind? Epinephrine beta or epinephrine alpha 1? GQ, yeah, epinephrine alpha 1, GQ, phospholipase C, IP3, DAG. DAG uses protein kinase C, yeah, not epinephrine beta. So don't mismatch and know the order, right? Insulin always and always decreases cyclic AMP inside your cell. Please know that, right? Yes, so the order is important. Protein kinase A is an allostric enzyme and its allostric modifier is cyclic AMP. I was asking you where is protein kinase C? It is with epinephrine epinephrine alpha 1 GQ. What is IP3 doing? Somebody said they don't know. I did signal transduction and they were blank when I said calcium calmodulin kinase. I hope you know IP3 calcium and calcium calmodulin kinase is in your lectures. Calcium calmodulin kinase is also epinephrine alpha 1 GQ. It's dependent on calcium. Yes, calcium calmodulin kinase is with GQ system, right? Does that make sense? Protein kinase C is also with GQ system. Protein kinase C and calcium calmodulin kinase are with GQ, not beta adrenergic. Then bring in your drugs. Also know that they have different effects on different cells. When you talk about albuterol agonist, smooth muscle cell relaxation, bronchodilator. Prednisone inhibits, again helps in bronchodilation, but it inhibits GI. Atroen inhibits GQ, same effect, bronchodilation. So you have to know what is GS, GI, GQ, then what drugs are acting. Otherwise, the whole connection will be lost. So bring in your drug and clinical application, at least the common ones right now, right? Epinephrine beta adrenergic is agonist, uses cyclic AMP, but have different effects on different cells. This is the summary. So let me ask you, GS, GI, or GQ, which is epinephrine beta adrenergic and glucagon uses? GS, GI, or GQ? Which one of them is used by epinephrine beta adrenergic as well as glucagon, GS? So you don't mess up. You don't mess up who is using which mechanism. So epinephrine and glucagon both use GS system and glucagon receptors are absent in the muscle. I want you to know growth factor in detail, not JAK-STAT, not serine kinase, no growth factor. When is RAS active? When bound to? When is RAS active? When is RAS active? When bound to GTP. Is RAS a monomeric G protein. It's not a trimeric G protein. When somebody was asking me what was the difference between cholera and pertussis, is it GTP, GDP? Please be mindful. The modification is by adding ADP. It's just making GS active and GI inactive. It's not acting on GTP and GDP. The toxins are modifying so that they are locked in one state, right? GRAS is with factor, persistent activation, cancer. Please bring that. Insulin model, what all you need to know. Insulin has growth factor-like activity, absolutely, but that's not primary. Insulin uses PI3 kinase and protein kinase B to do what? Tell me. Why does insulin use these proteins, PI3 kinase and protein kinase B? To do what? Why does insulin use PI3 kinase and protein kinase B? Anyone? GLUT4 expression, it's very specific. I have these two drawn together because PI3 kinase is not same as IP3. IP3 and PI3 kinase are different. We will connect why insulin increases GLUT4 expression. I think I will do that with digestion, which is the last segment. So PI3 protein kinase B is so specific, right? That is the mechanism used for GLUT4 expression. Triggers are very important. That will come up next time. Then you will connect signal transduction mechanism. Triggers are very, very important, believe me, to know which signaling is on at what time. But I will connect in the next week. Just do that for me. Which triggers increase which hormone? We will connect next. Please understand, you have to bring in your drugs too, yes? Cyclic GMP is with nitric oxide, protein kinase G is with nitric oxide. Don't forget to bring in your drugs and your clinical conditions. Always bring, and vitamins for this test. Drugs, clinical condition, and vitamin for this test. Carbohydrate digestion absorption is very lucid. Know the sources, know the enzymes, know only and only absorbable form is monosaccharide. Know the concept of reducing sugar in urine. Know all the transporters, sodium dependent, sodium independent. Sodium independent are all your GLUT. Please know all your GLUT transporters. Tell me which transporter is insulin mediated. Which transporter is insulin mediated? Which transporter is only insulin mediated? GLUT4, and the insulin uses PI3 kinase and protein kinase B mechanism there to increase their expression. You know proportional versus constant kinetics. You know ATP releases insulin. You know GLUT2 work with glucokinase. And 1 and 3 work with hexokinase, distinction. You know very well who relieves the transient hyperglycemia, which hormone relieves the transient hyperglycemia after meal? Which hormone relieves the transient hyperglycemia after meal? Insulin. Please know insulin function very well, which is in my next slide. You know lactose intolerance, bloating with floating, hydrogen breath test. Do functions of insulin. Good luck. If you have any questions, you can ask. I still get. Anyone has any question? I'm here.